In this module, I will discuss about the clinical trials. I will introduce you to this important important aspect of the biomaterials research, that is clinical trials. So, I will first try to refresh your mind that what we have learned so far. So, we have learned so far that in vitro studies that we have done we have learned to a greater extent. To the same extent or maybe little bit lower extent, we have also discussed that in vivo biocompatibility study, and in vivo is typically known as the preclinical study. Now, in vitro study, depending on the cell type, you need to have different assays, and different assays were also discussed, which can be used to quantify the cell functionality, cell viability on different material substrate. Now, as you go from or as you translate the in vitro results in the in vivo context or preclinical studies, you need to do experiments in the different animal models. So, in last couple of modules, I have discussed the need for the in vivo study. Essentially, in vivo study will have direct relevance or will have more appropriate relevance for that human environment. So, in vivo study is very important in the context of biomedical research, and it is called preclinical because it is the one step before the clinical trials, where human patients are involved to establish the efficacy of any biomaterial device or to establish the efficacy of any implant. So, coming to the difference, or if we can recall the difference between in vitro and in vivo. In the in vivo case, we have only one single cell type, unless otherwise the co-culture technique is used. So, this is a single cell type. And also the tissue remodeling aspect, the influence of hormones and the influence of dynamic change in the physiological environment. So, this dynamic change in physiological environment, all these aspects cannot be studied in the in vitro studies. For that, you need to conduct the in vivo experiments, where a material will be exposed to the dynamic change in the physiological environment. And what I have emphasized while discussing the in vivo studies, normally you have to start with the small animal models, and small animal models are very good for the toxicity study. Then you have to go to medium or large scale. And another things I have also emphasized during the discussion on the in vivo experiment or in vivo study is that the small animal experimental the results of the small animal experiments are to be validated at the medium or large animal model. So, and this ethical committee clearance is becoming more and more tougher as you go from the small animal model to the medium or large animal model. In the normal in vitro, you need to take the approval for the biosafety. So, there is an institutional biosafety committee. So, you need to take the approval from the institutional biosafety committee just to ensure that the way you conduct the in vitro cell culture experiments are actually as per the are actually following the ethical guidelines, so that you are not doing large scale manipulation to the cells as such. And this ethical committee or stem cell committee's approval is required in case you are using the stem cells in your research, like you are trying to understand that how stem cells will differentiate when they are being grown in a biomaterial substrate. So, in all those kind of cases, you need to take the institutional approval. But coming to the in vivo study, even if you use small animal, medium animal, large animal, irrespective of whatever the animal model experiments that you want to use, institutional approval or institutional IAEC, that is called Institutional Animal Ethical Committee's approval, is a must. And in all this animal institution animal ethical committee approval, you have to mention that how well you will be taking care of that animal's health during the implantation experiments and further. Now, when you go to the clinical trial experiments, then human subjects are required. So, clinical trial experiments that so essentially the small animal model and large animal model. So, the human patients who will take part in this clinical trial, they are referred in the published literature as well as in that many books and so on as the human subjects. So, that human subjects are involved in the clinical trials, and it is not only institutional animal ethical committee. So, both institutional and national ethical committees approval are required. Both institutional and national ethical committees approval is required for conducting prior to conducting the clinical trials. The other things is important, just before the start of the clinical trials, all the in vitro and in vivo biocompatibility results to be critically assessed. With clinicians. So, all these all the in vitro and in particular in vivo results, biocompatibility results are to be critically assessed, and National Ethical Committee's approval is required. In India, this approval is typically to be sought by DCGI, that is Drug Controller General of India, and that is that ICMR also gives this the approval for the clinical trials. So, we have one has to propose that the need for conducting clinical trials, and one has to also propose that how the results of the clinical trials will advance the scientific understanding on this particular set of new biomaterials or new implants, so that these materials finally can see the light of the day in terms of their commercial scale production or commercialization. So, this aspect is really important, and so that initial clinical trial plans need to be done in collaboration with clinicians, or in other words clinicians are to be involved in all these clinical trial proposal. Third important thing that in this clinical trial study is that you have to understand that what is the how many patients are to be recruited in the clinical trials, and these materials, whatever implants are to be used in the size and shape of device in the so implants are to be used in the same size and in the same size and shape of a device which are to be considered clinically. For example, let us say you have developed in zirconia and alumina based composites for femoral head or acetabular socket as a device. So, you have to shape this zirconia and alumina we cannot simply keep this zirconate and alumina a simple cylindrical piece or rectangular piece and put it in some human patients. It has to be first fabricated in the shape of the femoral head or acetabular socket, and then you have to put it inside the human subjects or human volunteers as part of the clinical trials. Or in other words, although your in vivo or preclinical study is conducted on simple geometrical shaped biomaterial implants, clinical trials must be conducted on the same in vivo tested material, but in the final shape and size of the device which would be ultimately implanted in the human patients. So, that point needs to be very clear. So, clinical trials is very important and very critical and very sensitive. So, therefore, all the serious approaches need to be taken during as required for the final commercialization of the biomedical device. So, having said all these things, so that major steps of clinical trials. So, essentially clinical trials are scientific experiments involving human subjects to establish the efficacy of certain biomedical treatment using some drugs or some biomedical devices or implants. So, first one is the design of the study. Second, so as I said, the design of the study is to be finalized with the help of the clinicians. Second one is the ethical committee's approval. As I said that in India, Drug Controller General of India approves this kind of clinical trials. Then clinical trial is registered, and then you can start your clinical trial and finally publish the results of the clinical trials. So, these are the steps. So, therefore, as you can see from this brief description of the major steps in the clinical trials, it is quite a challenging task and it requires very strict approval process at different stages. So, the types of studies and levels of evidence. So, clinical trials, one of the things that is very important is the clinical evidence. Many clinicians, when they do their higher studies or postgraduate studies or masters and so on, normally their projects mostly involve or their research projects mostly involve at the postgraduate level, mostly is the evidence based research. Like they do certain treatment and then they see what changes in the patient's health status, what changes those kind of treatment bring to the patient's health conditions, then they get the evidences, they record those evidences, then they analyze those evidences and then they try to explain it from clinical perspective. Now, it may not be necessary for clinicians doing postgraduate research to conduct lot of scientific or significant scientific experiments, significant scientific analysis, what a typically masters or PhD student at many of the academic institutions they typically conduct at the masters or PhD level. So, having said this, that clinical evidence is important in any kind of study and particularly for the clinical trials when it involves clinicians, because the clinicians believe in the clinical evidence. I mean, but as an academic researcher, one has to conduct scientific experiments, scientific analysis to prove certain point or to establish certain point. However, in the domain of the clinics or at the clinical settings, it is mostly driven by the evidence based research. So, they see something, they believe something and then they record those evidences and they try to infer based on those evidences that how this particular treatment can give patient satisfaction, whether this particular treatment can provide satisfactory patient's outcome. So, these are the kind of things that they normally do in the clinical setting. So, therefore, I just wanted to make you understand that these clinical trials, whole concept of clinical trials are to be thought in more from the clinical translational perspective rather than more from pure scientific understanding, more from pure scientific research, the way we conduct in typical academic settings, because academic settings and clinical settings are quite two different kind of concepts or two different kind of approach that we use while while conducting clinical trials. So, there are several kind of mode of studies, but one of the things that people do, and this is kind of a triangular kind of approach that we have sent. So, people read different editorials or expert opinion, clinical opinion, then they said case studies or case report. So, many times if you speak to any if you talk to any clinician in general in a hospital, they refer each time they conduct any operation, they say that we had a case study, or that means that the evidence that they collect from one operation on one single patient, they consider this is one single individual case study. So, like that if you conduct the similar treatment or experiments on 15 patients, you have 15 case studies for you to analyze the evidences that you can collect from 15 case studies. And then similarly, that clinical trials, as I will explain later, there is something called control trials and randomized control trials, which is most widely used in the typical clinical trials. So, as I said that in the clinical settings, research studies are to be can be classified in two modes. One is the observational study. You do something and you simply observe the patient's behavior. That does not involve any intervention or exposure. Intervention means the clinician does not need to intervene the treatment that the patient is currently undergoing. Their job is to just observe the patient's behavior or the patient's different organ functions and so on. Second one is the experimental study. Experimental study means, for example, in the case of drug trials, you give some drug A with certain dose X, but you just see that whether the dose X is quite high level dose or it can cause some side effect or it is causing more side effect, then you reduce the dose from X to X by 2, but then it requires intervention by a clinician or just changing the mode of the treatment or changing the treatment procedure itself, and then collect the evidences. In the experimental study, being a scientific researcher, you should be very much excited about that. One is called randomized study and second one is called non-randomized study. Randomized study means all the participants, like human subjects, they are randomly studied without any preference of any particular patient. So, this randomized study actually is very good in a sense that you do not have any bias for any specific patient, like you cannot choose all the male patients or all the female patients. It can be male, it can be female. You cannot choose that all the patients should be below the age 30. No, it can be random. It can be from age 15 to age 55 or age 25 to age 55. In this large window of the age, any patient who volunteers to take part in these clinical trials, they are most welcome and you must consider that. So, essentially to remove the bias, because that is very important in case of the clinical trials, you need to conduct this randomized control study. So, that we say that RCT, what I just mentioned one slide back, that stands for randomized control trial. Randomized means you do not have any control on the age of the patients, on the sex of the patients, on the previous history of the patients, but it controlled means then you have to see that how many number of patients, what is the different ages and all. Observational study, it can be more analytical study or descriptive study and so on, and there one of the studies sometimes people do is the cohort study, but our discussion will be mostly concentrated on this part of the slide, that is, it is experimental study, it is randomized study, and then how one can do RCT, that is randomized control study. So, as I said in the last slide, that experimental study essentially involves intervention or exposure, and it is non-randomized, and in case of randomized study, as I said, participants randomly allotted to study a cohort group, and it can be controlled or uncontrolled, but I will circle to put your attention on controlled study. So, there are different guidelines that need to be performed, since we are mostly interested in the randomized control study. So, this consort guideline is more important. Now, we should see what is consort guideline. Consort stands for Consolidated Standards of Reporting Trials. That means there are certain guidelines which are already mentioned in this particular website, that is www.consortstatement.org. So, this consort statement is essentially evidence based minimum set of recommendations of reporting randomized trials. It offers a standard way for authors to prepare reports of trial findings, facilitating their complete and transparent reporting and aiding their critical appraisal and interpretation. So, that means here all these interpretation that should be clinically meaningful interpretation. So, essentially you have to keep that clinical aspect in mind, and accordingly you have to prepare the clinical trial reports. So, the three pillars of clinical trials, randomized clinical trials, are: number 1 is randomization, number 2 is allocation concealment, number 3 is blinding. So, these three concepts I will try to take up one by one, and I will try to discuss in the next 3-4 slides that what is the meaning of randomization, meaning of allocation concealment, and third one is blinding. As I said, that these three constitute three pillars of randomized controlled trials. What is randomization? It is a process by which each participant, means here participant means that is a human subject, or people say human volunteer. Okay. So, here you use the term human subject, or each participant who voluntarily take part in this clinical trial. So, randomization means this is the process by which each participant has the same chance of being assigned to either intervention or control. What is the meaning of these clinical trials? Let us say you are using some femoral ball head, okay, that we are some people, some institution or some research groups would like to conduct clinical trials on the femoral head. So, and then there is a one type of femoral head which is very research grade, like zirconia, toughened alumina. Another type of clinical head is stainless steel clinical head or cobalt chrome moly clinical head. So, femoral head. So, one side it is ceramic and one side it is metal. So, a human participant or human subject or human patient would not be knowing whether he or she is getting ZTA or SS. They would not be, so each patient would have equal probability to receive either ZTA or metallic femoral head. So, randomization is effective in reducing bias, as I said before, because it guarantees that treatment assignment will not be based on patient's prognostic factors. So, this is very important, and investigators also cannot favor one treatment group over another by assigning patients with better prognosis to it either knowingly or unknowingly. And procedure selection bias has been documented to have a very strong effect on outcome variables. So, sample randomization, for example, like 24 patients are there in a simple clinical study, and these 24 patients they have like assigned like A, B, A, B, B or something like that. So, it's basically when you toss a coin, so you have equal probability either it will be head or it will be tail. So, head is like treatment, tail is like placebo. So, you essentially here it has been methods has been mentioned, you toss a coin, random number table and computer generated random numbers. So, tails is here control or placebo and heads is their intervention. So, the control things, if we go back to the same thing, so let us say this is your experimental grade femoral head, zirconia, toughened alumina, which you have just fabricated in the laboratory scale, but commercially and clinically stainless steel femoral head has been used for ages. Now doctors are trying to do clinical trials, or researchers conducting clinical trials to see whether zirconate and alumina can potentially replace stainless steel based femoral head. So therefore, if you put head and tail, so if head is ZTA, tail can be stainless steel. So the patient does not have any chance of choosing whether they will get zirconate and alumina femoral head or whether they will receive stainless steel or metallic femoral head. So therefore, that biases can be removed clearly by this randomization process. Second one is that allocation concealment. Concealment to keep something secret. So like a patient is now allocated either ceramic head or metallic head, right. Now patient would not be knowing what is the femoral head composition that he is getting or he or she is getting. That means patient will be knowing only that they are getting one potentially clinically acceptable femoral head. So allocation concealment refers to the technique used to implement the sequence, and allocation concealment mechanism aims to prevent participants or recruiters from knowing the group to which the next participant will be assigned. For example, there is 24 patients, as I said in the 1 or 2 slides back. So if the patient A is getting one type of head, patient A will not be knowing what patient B will receive, because all these femoral head in the context in reference to femoral head description. So there will be no clarity what each of the patients will be receiving. So sequentially numbered opaque sealed envelopes, like in case of drugs that has been mentioned, it is pharmacy controlled, numbered or coated and all. Now third one is blinding. Blinding means it is essentially masking. Unawareness of the study group to which trial participants have already been blinding. So purpose of blinding is again bias reduction. Each group blinded eliminates a different source of bias, and blinding is most powerful when there is a subjective component of treatment or evaluation. What it means is that a group of patients will be receiving, again I go back to my original example of this femoral head. So a group of patients will be receiving one type of ceramic head, another group of patients will be receiving another type of femoral head. So each group they will not be knowing what kind of femoral head that they have received. So this is that kind of examples that I thought that would be interesting. And then as I said, that ethical approval is a must, and all research involving human patients should be conducted in accordance with the 4 basic ethical principles. Number A is that autonomy or respect for person or participant, that means one has to fill up the consent form. Each human subject, each human volunteer taking part in the clinical trials must fill up the consent form. That means they are willing to participate, and they are willing to participate in a very honest manner to take part in this clinical study. Beneficiaries like provided written details of the study and encouraged to ask questions, like each human patient is encouraged to ask as many questions to the investigators possible. Third ones, that there is no harm, non-maleficence, so that provide written guidance to the care of the patients. The patient care is important, and which is equally important when you conduct the preclinical trials in the in vivo experiments. And fourth one, justice, selection of participants like randomization process is to be ensured. The other things that I have mentioned here, that every human or animal study need institutional ethical approval as well as ethical in the multi-center approval. So there is something called single centric approval, and there is something called multi-centric approval. So single centric approval means, for example, you take any hospital A, you want to conduct the clinical trials on the limited number of patients on single hospital, then you call single centric. Multi-centric means you need to conduct the clinical trial experiments on a large number of patient population, but not restricting to one single hospital, but again in 3-4 different hospitals, perhaps in different parts of the country, same country. So, therefore, this is like multicentric study, and that against approval from all the participating hospitals which will participate in this study. There is format for ethical approval which may differ slightly depending to the infrastructure, and ethical committee approval number and soft copy is mandatory during submission to the journal. So, the other things that is because in the clinical trial, one of the thing that legality comes into picture in a big way, because you are using human subjects in your experiments or in your scientific study. So, it is illegal to perform a trial with a medicinal product without having acceptance from your national competent authority. This national competent authority, I have mentioned it before, that is called DCGI, the Drug Corporation of India, that is very important. And proposed trials should be carried out only and is necessary under Schedule Y of the Drug and Cosmetics Act, and also there is a new drug approval. So, what happens in the context of biomaterials, this since that biomaterials and implants, the clinical trials are something which has becoming or which is becoming popular. So, essentially people follow whatever drug related trials that ethical committee's approval is there, similar ethical approval is also followed in case of the biomaterials. So, I think I will stop here and I will try to complete on these clinical trials with some more discussion, so that you understand the fundamentals and other concepts on these clinical trials.